introduce myself and my colleague. I'm Dr. Tabinda Hasan and my colleague Dr. Kavita Ganesh. We are in the anatomy department of Princess Mira University in Riyadh, Saudi Arabia. And I will be talking about the proteins and the genes that play a role in the spread of Alzheimer's disease. So first of all, we'll talk a little bit about the Alzheimer's itself. Alzheimer's disease is a progressive disorder where the brain cells waste away and they degenerate and die. So it's the most common cause of dementia, which is a decline in memory and cognitive functions. There is a typical patient of Alzheimer's would present a continuous decline in thinking and behavioral and social skills that disrupts the person's ability to function independently. So what happens is that the decline in the functions reduces that person to a subhuman level. So you would see a person who has Alzheimer's to appear like demented or a little bit lesser than the normal humans. So that's why in the common English language we use the term demented, which is supposed to represent somebody who is stupid or foolish or doesn't understand things. So this is a typical picture of the demented person. And that's what happens in Alzheimer's. And that's why it's one of the most dreaded diseases that we can ever encounter. I would like to go back to slide number 2.
Okay.
I'd like the help of the moderator, please, to bring me back to slide number 2.
That one, right? How does?
Yeah, the second one after the title slide.
Okay, okay.
I guess it's moving on its own or something. Yeah, this one. So we see the normal healthy human brain, as you can see, has billions of neurons that are shown in this picture with the bright red color. So these billions of neurons, they are interconnected with each other in a normal human brain. And these neurons, if you look at a particular neuron, if you see in the lower part, in the lower side of the picture, you see a particular neuron, it has got a head and it has got some processes that are like arms that they are called dendrites, and then there is an axon. This is a general structure of a neuron for those who need to know a little bit about the structure of neurons that interconnect together. So once again, you can see on the picture on the right side, there is the picture showing the interconnected neurons. So now we move on to the next slide. We see how the Alzheimer's disease affects the brain. So what happens in Alzheimer's is that it disrupts the networks between the neurons. The neurons are the ones that generate electric signals within the brain, and then these electric signals move within the brain from one area to another to bring about the normal function of let's say, a cognitive brain, a cognitively effective brain. But when these networks are disrupted, it is something like if you connect 2 or 3 pieces of electric wires together to light a bulb at one end, and even if one of those wires is disconnected, then the bulb at the other end would not light up. The function would not occur, the result would not occur, and that's what is happening in Alzheimer's. The connections between the neurons is disrupted. So the cognitive functions will start disappearing. So what I said was the lighting of the bulb, that would be affected. So first of all, memory would be gone. So in any patient with Alzheimer's, the, the first thing that is affected is loss of recent memory. So they start forgetting the recent happenings, and you would typically, Alzheimer's actually doesn't have any physical symptoms. So the patient, the person initially in the initial stages of Alzheimer's is very much like me and you. There are no symptoms. So the person complains that I have become very forgetful. So this is a usual. They would place their keys somewhere and forget about it. They would the most common is forgetting of car keys, forgetting of house or office keys, forgetting the mobile somewhere where they kept it. So it starts like this, loss of recent new memory. And later on, the most saddening part of the disease, the most, in my mind, I think the gravest effect of the disease is when there is loss of old memory. And loss of old memory includes loss of relationships, because it's there with us since the start. So when you forget what was your relationship with your spouse or with your children, and these patients, they stop recognizing their relatives, even their husbands or wife at a later stage, their children. So that is the most sad part of the disease, and that is what alienates and separates a patient suffering from Alzheimer's from the whole world, because they live in a bubble. They are separated from the whole world because they cannot connect anymore on an emotional level. And in the end, the last stage that is affected, the last stage is that they lose their language skills. So they forget how to form words, just like children. So they would have the idea in their mind, but they don't know how to present it verbally. And they lost the power of reasoning and of social behavior. They also forget how to do the day-to-day daily tasks, like, for example, bathing themselves. How would they bathe themselves? How would they clothe themselves, button their shirt? So these are the day-to-day activities, tie their shoelaces. So all of this is forgotten. Even their favorite games, they don't know how to play it anymore. So eventually a person with Alzheimer's becomes very helpless, and he is unresponsive to the world outside. So we move on to the next slide now. Now that we know what all happens in Alzheimer's, like, it's a very natural question to wonder about, why did it happen? And why doesn't it not happen to everybody? Why only some people? I would like here to tell you something very interesting, that these days, this is the era of molecular medicine, and there are a lot of genome-wide assays, or let's say gene-related studies. Often there are studies involving the whole population where they want to study a particular genetic trend. In those cases, the rule says that if the researchers by chance discover the gene related to Alzheimer's, the one that would cause Alzheimer's later to that person, it is recommended in the medical ethics to not inform the person about that gene. Why is it so? It is so because the doctor informing the person that you will develop Alzheimer's, let's say after 10 years, it would not change the possibility of the occurrence of that disease or the course of the disease in any way. And also, there is no cure for Alzheimer's. There is nothing that we can do to prevent it or to substantially delay its progress. So in such cases, it is considered ethical to not inform the person of that impending doom. So the medical ethics says that if in genome-wide assay you accidentally discover an Alzheimer's gene, you as a researcher are not supposed to disclose that information to that patient. So this is one of the really interesting things that I found about the Alzheimer's disease, because usually the medical code of conduct says that if the researcher discovers something like, for example, a malignancy, it's their duty to inform the patient and the physician about this. But in Alzheimer's, no, we are not allowed to inform. So what are the main characteristics? What causes this disease? First of all, there are the formation of plaques. So once again, I would like you to look at the picture. You can see a lot of neurons. The dark colored neurons are there. Now these neurons, like I gave that example of electric wires, these are the ones that conduct electrical signal, or you would say neuronal activity. So these neurons, they need to be not insulated. Like, for example, if there is an electric wire and you insulate it, that would interfere with the conduction of the signal itself, the transmission. So these neurons need to be in a very clear, clean environment without any foreign materials around them that would disturb the activity of the neurons and the transmission of signals between one to the other neuron. Now in the picture, once again, you can see the brown circles. These are the deposits of an abnormal protein that is called the beta amyloid protein. This beta amyloid protein, especially the 42 type, has a very strong tendency to clump together. So these proteins are like sticky materials, and they clump, they stick to each other, and they also stick around the neurons. So they form sorts of insulated barriers in the brain tissue. And these barriers, they physically interfere with the transmission of the signals. So these are rightly called the choke cells. What is the meaning of the word choke? Choke means when you don't allow a person to breathe, you literally strangle that person. So here what is happening is that the neurons are not allowed to, let's say, breathe, let's say, do their normal activity, which is the transmission of signals. So they are being choked. So these are due to the abnormal clumps of protein, and they have a special name. This clump, that abnormal clump that is found, is called the tau proteins. So the tau tangles, they are very typical of an Alzheimer's brain. They are not found in the normal brain. This is a protein that would never be found in the normal brain. The tau tangles, they tangle, like they stick to one another. So they are called the tangles. So the tangles of tau, they block the neurons transport mechanism, and also the signals at the synapses. So the transmission of signals from one neuron to another, it would be interrupted. So overall, this loss of connections between one nerve cell and another is the main reason why all the cognitive capabilities of the human brain decline. So what all we discussed earlier, like there is loss of memory, loss of function, loss of skills, and all that is because of the abnormal deposition around the neurons that interferes with their capability to act as a neuron. So there are, like I mentioned before, there are abnormal deposits of beta amyloid protein and the tau protein. So in the final stage of an Alzheimer's disease, the brain tissue significantly shrinks. So in the picture that you can see, on one side you can see the normal brain, which is, of course, it's plump, it's larger in size, and it has the gray matter and the white matter. We will not go into the details of that. But if you compare it with the diseased brain of Alzheimer's on the left side, you see it's so much smaller, and also the gray matter has significantly reduced. The gray actually doesn't appear gray, but it appears pink, as you can see in the picture in the normal brain. And the white matter does appear white. So you can see there is an abnormal shrinkage of the amount of gray matter, which makes the white matter appear to increase. Actually, the white matter also has not increased. Both the gray matter and the white matter have reduced, but the significant reduction in the quantity of gray matter as compared to the white matter, which is not that much affected, that leads us to visually have the impression that the brain only has white matter, all the gray matter is gone. But actually both of them are affected. And because in the gray matter lie all the functional areas of the brain, the area for thinking, the area for personality, for all the motor, the sensory functions, for hearing, for speaking, and also for vision. All of these areas are lying in the gray matter. So obviously, if that gray matter is significantly destroyed or reduced, then all of the functions that I mentioned earlier would be affected in a negative way. So now we come to the question once again, like I was saying, why does Alzheimer occurs? What causes this disease? So early onset Alzheimer's is when it occurs at a significantly early age as compared to what we were expecting. So normally we expect the Alzheimer's to be occurring in old patients, which is in old persons, and it's true. Like people in their 60s, in their 70s, these are the ones that usually have Alzheimer's. And here I would like to mention one thing. So many of the cases of Alzheimer's, they go undetected. Like the old people around us, our grandfathers, grandmothers, they are, many of them might have been Alzheimer's patients and we did not know because let's say 10, 15 years ago, 10 years before or 15 years before or 20 years before, there was not so much awareness about Alzheimer's. People did not know, the common man did not know what is Alzheimer's disease. So the thing that was taken as part of old age, that senile dementia, might not actually have been senile dementia when the old people used to fail to recognize their relatives, they couldn't dress themselves, they were forgetting, they couldn't speak, they couldn't eat on their own, and that all would be attributed to the senility of the old age. But it's not. Maybe they were Alzheimer's patients, full-blown Alzheimer's patients. We didn't just know about it because this term in medicine is still a new term. I mean, still researchers are in their, let's say, in their adolescent stage when it comes to Alzheimer's. We still are, there are a lot of things that we don't know about Alzheimer's. So early onset Alzheimer's is when it occurs before 60 years of age, and the earliest known cases are in their 30s. So this is caused by, of course, genes are inherited, but Alzheimer's is a disease that needs two identical sets of genes to manifest itself in the early onset. So when there is a dominance, when there are two genes, so it's dominantly inherited Alzheimer's. And this is of course caused by genetic mutation. And I would be talking about genetic mutation a little bit later. I would explain in more detail. So there are chromosomes that are there responsible for the mutation. There is mutation or an alteration in the chromosomes. There is chromosome 21 mutation, there is chromosome 14 mutation usually, and chromosome 1 mutation, although these are not the only chromosomes that are mutated. So in the chromosome 21 mutation, there are abnormal amyloid precursor proteins. So when these amyloid precursor proteins are present in large amounts, of course, they would lead to lots of amyloid bodies in the brain. In chromosome 14 mutation, there is abnormal preselin 1 gene. And in chromosome 1 mutation, there is abnormal preselin 2 gene. So I would discuss a little bit about these. So let's start with the amyloid depositions. The amyloid depositions are like sticky insulated material between the neurons. And now when we come to the chromosome 14 mutation, that is the preselin, the chromosome 14 and the chromosome 1 mutation. So we will talk a little bit about the preselin 1 and the preselin 2 genes. Alzheimer's, as we all know, is the most common form of dementia. Mutations in the genes that encode the preselin 1 and the preselin 2 and the amyloid precursor proteins have been identified as the main genetic causes for familial Alzheimer's disease. So once again, please note that Alzheimer's disease runs in families because, of course, anything that is genetically encoded would run in families. To date, until today, more than 200 mutations have been described worldwide, means 200 types of mutations have been described worldwide when it comes to the preselin 1 and the preselin 2 genes. Now, these two genes are very highly homologous, they are similar. The mutations in preselin 2 are relatively less as compared to the frequency with which the mutations in preselin 1 have been reported. When we look at medical literature and we look at the review studies describing the mutations of these genes, then preselin 2 mutations are more common in the European and the Korean and East African population. And interestingly, preselin 2 mutations appear not only in Alzheimer's, but also in patients with other disorders like frontotemporal dementia, the dementia that is related with Lewy bodies, even in breast cancer and cardiomyopathy, Parkinson's disease, etc. So these genes, please note that these genes are not only uh, typical of Alzheimer's, but also they would be found in other medical conditions, as I just mentioned. So now we come to the late onset of Alzheimer's. The late onset Alzheimer's, as I mentioned, would be appearing in patients that are more than 60, over 65 years of age, typical old man. You see the picture, that's the picture of a typical Alzheimer's patient, old, lost in his own world, unaware of what's going on around him, completely cut off from his surroundings. So that's late onset Alzheimer's. So the evidence of the hereditary component is too much in Alzheimer's. You cannot separate Alzheimer's disease from genes. Of course, there are environmental and lifestyle factors that might contribute to the risk of developing late onset Alzheimer's. But once again, the research is very, let's say, in the juvenile stages. If I say smoking predisposes a person to Alzheimer's, or unhealthy diet predisposes the person to Alzheimer's, like, I would not be right, because there is no very solid proof or lots of evidence in medicine. Yes, there are hints about lifestyle factors and environmental factors, but we cannot say anything definitive until now. So we move on to the next slide. Now we will talk a little bit more about the apolipoprotein E, that is called the APOE gene, and its role in the late onset Alzheimer's. The APOE gene is a special gene that is found on chromosome 19, and it has 3 phenotypes. Means it has when we were students, we used to call phenotypes as the phenotypes normally means how a person looks like. So I have a particular phenotype. So in this gene, there are three physical manifestations of this gene, which we call the 3 alleles. So there is allele 2, 3, and 4. But the allele 4 is the one, which is the typical, that is related with the APOE gene and the most established genetic risk factor for late onset Alzheimer's. So the APOE allele that is found in only 10% of the population, but see, note the statistics, it's found only in 10% of the population, but it increases the risk of Alzheimer's to 8-fold or up to even 10-fold. So some people with the APOE4 allele, they never get the disease. So when we say up to 8-fold, it's still not 100%. So it's not necessary that every Alzheimer's patient, especially in late onset Alzheimer's, would be having, and every Alzheimer's patient with the late onset Alzheimer's 100% would be having an APOE allele 4 gene. But not every person having an APOE allele 4 gene would invariably develop Alzheimer's. So it goes like that every patient has that gene, but not every person having that gene becomes a patient. Now we move on to some of the new, now as the researchers have been done, genetic researchers and genome-wide assays have been done to analyze the DNA of populations. So there are more varieties of genes that have been discovered, not just the APOE gene, but also other genes that have been discovered to be related with Alzheimer's disease. Of course, the single most common risk gene is the APOE gene, but there are other new genes that are, at this stage, they are being called as risk genes. We don't name them causal genes, because we are not sure if they really cause Alzheimer's, but we would still uh, give them the term of risk gene, because the presence of these would predispose to development of the condition later on. So we have certain, I have made a list down of all those genes. So the more prominent ones are the HLA, the PTK, the DSG gene, the CASS, the FER-M2. These are just the names of the genes. So let's try to understand a little bit more. Now we have been talking a lot about Alzheimer's and the genetic basis of Alzheimer's. Let's talk a little bit about what are the biological processes that just go haywire, that are disturbed, that go awry in Alzheimer's. So what is different between a normal brain and an Alzheimer's brain? Of course, at the functional level. Amyloid metabolism. In the Alzheimer's brain, this is highly disrupted. It is slowed down, and all the amyloid that is present is not metabolized, and that's why it's not removed. So there are amyloid deposits, and these amyloid deposits form physical barriers that interfere with the transmission of the neurons, and that's what Alzheimer's is all about. Alzheimer's means reduced capacity of the neurons to transmit signals because of physical barriers. These physical barriers are in the form of amyloid proteins. Then, of course, certain immune responses of the brain, for example, the microglia, these are very strongly associated with Alzheimer's. So when there is disruption in the function of the microglia, there is then it leads to Alzheimer's. It has been seen that proliferation and activation of microglia in the brain, especially concentrated, especially the activity is found more around the amyloid plaques. So this is a prominent feature that helps us to identify the Alzheimer's disease. Abnormal functions of the microglia and increased functioning, abnormally high functioning and proliferation of the microglia cells. Microglia cells are cells that are found in the brain, and they are mesodermal in origin, and they are phagocytic in function. So their normal function is to eat up or to phagocytose the foreign materials, the unneeded materials that are found in the normal brain tissue. But of course, they are not doing that. So that's why the immune responses, because a microglia is part of the brain immune system, so that is interfered with. Then we have abnormal protein degradation. Cellular protein degradation is slowed down. So of course, when the protein degradation is slowed down, there is higher levels of protein than needed in the Alzheimer's brain. Then I would like to talk a little bit more about the microglia function. As I mentioned before, that the microglia activity, its proliferation is increased and concentrated amount around the amyloid plaques. And this is what helps us to identify the Alzheimer's brain. Human genetics data point to a key role of microglia in the pathogenesis of Alzheimer's disease. The majority of risk genes for Alzheimer's disease are highly expressed, and many are, please note, selectively expressed by microglia in the brain. So microglia do have a real-time role in the pathogenesis of Alzheimer's. There is mounting evidence that microglia protect against incidence of Alzheimer's disease. This is what the researchers proved before, that microglia are actually protective. Yes, they are part of the brain's immune system, but at a certain stage, they start overacting. So this is what we are trying to say. So impaired microglial activities and alteration in the microglial responses to the beta amyloid protein are then associated with increased risk of Alzheimer's. There is abundant evidence that activated microglia can be harmful to the neurons. So these microglia, the same microglia that were supposed to be protecting the neurons, when they are proliferating and hyperactive, they actually don't protect, but rather they damage the neurons. They can be harmful to the neurons. The microglia mediate synapse loss. What is the most important part of a human brain that is functioning is the proper healthy synapses, the interconnections between the neurons that allow for signal to transfer. So when the microglia, they attack the synapses, they cause synaptic loss, they even engulf or eat up the synapses. Just like, like a complement dependent mechanism. So it's like a self-feeding cycle. They damage, and the damage promotes more damage. So it's an unending cycle. They can also exacerbate tau pathology and secrete inflammatory factors that can injure neurons, either directly or via the activation of neurotoxic astrocytes. Astrocytes are another type of cells. They are star-shaped cells also that are present inside the brain. So gene expression profiles, these studies, if you go across these studies, they show the microglia in multiple states of activation in the neurodegenerative disease setting. And this explains the disparate role of microglia in the development and progression of the pathology of Alzheimer's. So microglia were supposed to be protective, but when they start over-functioning, they are disruptive. So we can see that there are other types, apart from microglial genes, there have been other types of genes that have been discovered, like the tri-OB gene and the TREM2 gene and the CD33 gene. So these are some of the newer genes that have been discovered. So now we come to another type of gene, that is the ATP binding cassette. So we go to the previous slide. The ATP binding cassette, or the ABCA gene. So before I talk about this, let's understand what the ATP binding cassette is. In Alzheimer's disease, which is a progressive neurodegenerative disorder, and actually it's the most common form of age-related dementia that begins with memory loss and progresses to include severe cognitive impairment. So this is what is happening in Alzheimer's. Let's see why it's happening, why in keeping the ATP binding cassette in our focus. A major pathological hallmark of Alzheimer's disease is the accumulation of beta amyloid peptide in saline plaques in the brain of patients. The exact mechanism by which Alzheimer's disease takes place still remains not fully known. However, an increasing number of studies have suggested that the ATP binding cassette, which is, you can see in the figure on the side, these are the ABC transporters which are located on the surface of the brain endothelial cells of the blood-brain barrier and the brain parenchyma. This ATP binding cassette may contribute to the pathogenesis of the disease. Recent studies have unraveled important roles of ABC transporters, including, now let me name some of them, ABCB1, which stimulates P-glycoprotein, ABCG2, which is also present in the in breast cancer, which is also called the breast cancer resistant protein, the APCC1 in the pathogenesis of Alzheimer's disease, and the amyloid beta peptides deposition inside the brain. Therefore, understanding the mechanism by which these transporters contribute to amyloid beta deposition in the brain is important for the development of new therapeutic strategies against Alzheimer's disease. So I have tried to summarize this information in this slide. We see that the ABC proteins, they have the subfamily, we call them together ABC, but they have subfamilies, family of A, B, and C separately. They are highly expressed in the brain tissue, the parenchyma, and the endothelial cells of the blood-brain barrier in the Alzheimer's brain. These ATP binding cassettes have a role in cholesterol and lipid metabolism. So actually in the brain, when the cholesterol and the lipid metabolism would be hampered, the condition of Alzheimer's would be exacerbated. The ABC transporters contribute to beta amyloid peptide deposition inside the brain, which is the hallmark in the pathogenesis of Alzheimer's. So everything in Alzheimer's, let's note that it's just increased deposition of proteins and plaques and let's say amyloid proteins and lipid plaques, and that's what it is all about. So it's the normal brain, the normal neurons, but they are being coated by abnormal insulatory material, and this is what is Alzheimer's. Move on to the next slide now. So how can you diagnose this disease? Before even starting on this, I would say it can only be safely diagnosed after death, because it's not possible to there is no specific test like like a blood test that shows certain types of cells going up or down, certain levels going up or down. So the only real diagnosis is after that post-mortem. You would see the brain significantly shrunken in size, but then there are tests which are the symptomatic tests. You would see the functional aspect of the patient, and on that basis you can functionally diagnose. So if we take a medical history, so of course the patient, since now we are branding that patient as having forgotten many events, so the history is never taken from the patient himself but from the caregivers. We ask about the person's day-to-day function and behavior. We ask certain questions like, uh, does he or she forget a lot? Are there sudden mood swings? Is there a lot of depression? Is there a lot of shouting? Is there a sudden change in behavior? So Alzheimer's, the personality typically changes. The caregiver would tell you he's not the same person anymore that we knew. So the personality totally changes. Then there are certain tests, the basic tests like memory tests, and then tests for language skills, like we show them a picture and then we ask them, what is this? Can you recognize this? So you show them an apple and they need to formulate the word apple. But in the case of Alzheimer's, the patient might remember that it is something that he has seen, but he no longer remembers the name for it. So it's something like that. So when you see a patient who cannot name the basic objects, then you have a strong diagnosis pointing towards Alzheimer's. In the brain scans, some of the areas are, especially now in the color Doppler scans, that are the colored scans, it's very easy to measure in terms of the color. The areas that are functioning more as compared to the areas that are less active. So the Alzheimer's, typical Alzheimer's brain is overall less functional as compared to a normal brain. Then there could be biomarkers in the CSF and in the blood. Especially you could find elevated tau proteins in the blood, but this is not always there. So this is like a questionable test. So that's why we say that the most definitive diagnosis of Alzheimer's would only be after death. Now we move on to the next slide. We see how is Alzheimer's treated. To be very frank, there is no cure for Alzheimer's, and the disease just naturally progresses until it reaches the terminal stage. But there are certain therapies that could be given as palliative care to just reduce the severity or the effects, or the let's say the bad effects of the condition or the symptoms. Nowadays, there are certain drugs, but still under trial. They are designed to preserve acetylcholine in the brain, which is a neurotransmitter of the brain. And it is supposed that these drugs that preserve acetylcholine would enhance the memory, the cognitive skills. So there are certain drugs, but once again, they are not the drugs to cure the disease. They are just drugs to make the disease a little bit mild. Non-drug approaches like behavior cognitive therapy, these are there. Meditation. I think the speaker after me would be talking about from what I read in the program, the non-drug approaches like the spiritual aspect. So once again, like I would just touch it a little bit because there is another paper that would be dealing in more detail about it. So the behavior is like summarized in that term the cognitive behavior therapy. So this is the one that is usually done. We try to change the environment into a more positive one for the patient because of the mind-body therapy principles. Then lifestyle modification, of course, in any disease. The patient needs to maintain a healthy diet, exercise, should reduce stress, have sufficient sleep. But of course, like I said, there is no specific cure for the disease other than just trying to give palliative symptomatic treatment. Now we move on to the next slide, identifying risk factors for the cognitive decline in Alzheimer's. So what are the risk factors that lead to this Alzheimer's? You would, even before that stage comes when you would want to do a genome assay or study the genes, or maybe hope for finding an abnormal gene there or an abnormal protein plaque or tau protein, or like all the things that I mentioned. Even before that stage comes, first of all, that stage is not, these are just research-based facts. But actually, in intervening in a patient to find all that, what I just mentioned, it's there. But as a doctor, you cannot find it because there are no specific tests that you could order to detect those genes or those plaques in the brain or those things. But yes, researchers, they confirm this fact. But research is different from everyday clinical practice. So you couldn't possibly order a test to find out all that I mentioned. But in the history taking, you would find certain telltale signs like change sleep patterns. So there are problems with sleeping. The person says that he is awake most of the time. Then there is psychosis. There is abnormal change in personality, or there is disturbed behavior patterns. There is hallucinations. The person would imagine more towards an abnormal level. There would be delusions, and the person himself would report of mild cognitive impairment. So we call that term self-reported mild cognitive impairment. So it's something like the patient would say, my brain has seemed to slow down. I seem to take time in processing normal information. I think my brain is not that sharp as it used to be. So this is the way in which the person would express himself. So this is self-reported mild cognitive impairment. So once again, it's reported by the person himself, or maybe the caregiver, the primary caregiver. An interesting finding in most of the patients of Alzheimer's is very high blood glucose levels. So when we try to correlate it, like why would the blood glucose levels be so high in Alzheimer's disease? Actually, Alzheimer's is an ongoing pathology in the brain. And whenever there is any pathology, it puts the body to stress. And what is the direct result of stress? High cortisol levels, and high cortisol levels in turn leading to high glucose levels. So no matter if I advocate a healthy lifestyle to you and a good diet and exercise and all that, I would still not be making my point here. To be realistic, age and genetics are the best known risk factors for Alzheimer's disease. So if you carry the right genes, when I say the right genes, I mean those genes that are markers for Alzheimer's. If you carry that genes, and if you are of the right age, means if you are above 65, then obviously you would be the best chosen candidate for Alzheimer's disease. Now we move on to the last slide. So whatever I said just now, it just makes us wonder about some things. What at all makes this disease process begin? We talked about abnormal protein and plaques, amyloid plaques and all that, but why does it start in a normal brain? What actually triggers that process? And why is it that some people with having the same genes, they would develop Alzheimer's, and while others would not? So when we were talking about the APOE and the ABCA1 genes, I made a point there, and I would repeat it now, that all the Alzheimer's patients have those genes, but not every person having that gene would develop into an Alzheimer's patient. So this is a research question. All these questions have still not been answered. Then how the genetic factors interact with lifestyle. This is also a very interesting question. If there are genes, you would say, if there are genes, okay, I would get Alzheimer's if I manifest these genes. But then some people don't manifest it. So the question goes back to, were they having a different lifestyle pattern, or were there environmental factors that were different between two people, leading to one developing the disease and one not having the disease? So what is the exact interaction between nature and nurture? What is the exact interaction between genetic factors and the lifestyle and environmental factors? And then how does all of it vary from one person to another? Even the onset of the disease and the severity of the disease is different from person to person. And until now, we don't have the answers to it. Why? So in the last slide, of course, this is my last slide, the next slide, I would like to give, just for people who are more interested in this topic, there are certain links that you could follow. I hope these slides would be there available for people to download, for everybody, for the participants. So you can read up a little bit. These are very interesting projects. There is this Alzheimer's disease sequencing project that is being done by the NIH, that is the National Institute of Health of US. And then there is this Alzheimer's disease genetics consortium. Here you would find a complete database of real patients and information pertinent to Alzheimer's. Then there is this late onset Alzheimer's disease genetics study. And there is once again a very interesting genetics of Alzheimer's data storage site. So here, once again, for those who are more into the genetic aspect of the disease, they could find a lot of interesting information here. And then there is the National Cell Repository for Alzheimer's disease. This is a national resource that helps researchers find genes. So as a researcher, these links would be very useful to you. So if you are interested into that, exploring more about Alzheimer's, especially the genetic aspect, then these are the resources that maybe you could have a look at. So that's all. I want to conclude my presentation now. Thanks a lot for listening.
Thank you, Dr. Tabinda, for presentation. If you have any question.